To refresh ourselves on the cardiac muscle histology, which we've covered this back in AP1, but in case you're not remembering every bit of it, let's go through it a little bit. You're going to have these cardiac muscle cells. They're shorter than the skeletal muscle cells. They have striations, those little lines going across. Then they have those thicker lines at the end known as the intercalated discs. That helps to hold the actual cells together. Now, they are on the shorter side. The cells are a little bit fatter. They're interconnected. So hopefully these might be kind of triggering and thinking about a few things back from AP1 histology. You're going to have this connective tissue matrix. What it does is it's having the fibers, kind of like this skeleton fiber, that's helping to hold all the different cells together, creating more of a cohesive mixture of everything to keep it together and prevent it from pulling apart. Now we have T-tubules. Now hopefully remember, T-tubules, also known as transverse tubules, are what help to take the signal from the surface and carry it deep down into the actual cell. Now, they're not as many as the skeletal muscle, but they are wider to carry more information or more ions. The SR, or sarcoplasmic reticulum. Now, it is simpler than skeletal muscle, but still functional. And mitochondria. Now remember, mitochondria are what create the power, the energy for the cell. The mitochondria can take in the products of your carbohydrates and then convert them into ATP, so adenosine triphosphate. Now notice, 25 to 35% of the cell volume is mitochondria. Now, you don't need to memorize percentage. That means a quarter to a third of the entire cell is just mitochondria. Why would you need so many? Because those cardiac muscle cells are constantly working. They don't take breaks. You can't take a break to re-energize or to build energy back up. It has to keep going. So you have tons of mitochondria that allows you to keep perpetually making energy so the cells keep working. Now I mentioned those intercalated discs before. Just remember, that's what holds the cells together. Now they anchor the cardiac muscle cells one to the next, preventing them from separating. But they also have these desmosomes. Now desmosomes, think of them almost like Velcro, helps to really kind of keep those cells together, making sure that those intercalated discs don't pull apart and tear. The whole purpose here is to keep those cardiac muscle cells together and functional. You'll also have gap junctions in there. Now, gap junctions are kind of like pores, openings that allow ions to travel through. This is important because these cells allow ions to travel through, but those ions allow for it to be electrically coupled. Because your cardiac muscle cells, your heart, has to be able to beat on its own, has to be able to carry information through the heart to know how to coordinate its heartbeat. These cells, this gap junction in between the cells, allows that to occur. It allows each adjacent cell to have communication with the one next door. So that's the ions passing through. So if we're looking at it this way, up top, this is the basic drawing of cardiac muscle cells. You'll notice if you look right here, that's showing the desmosome, kind of that almost like Velcro-like sticking. That's helping to keep the actual cells anchored together and make sure that those intercalated discs don't pull apart. It's allowing for also, you have those gap junctions then, which allow the transmission of ions from cell to cell. So adjacent cell to adjacent cell. It's all about how the functionality, building up to the physiology of how these cells work. Because the anatomy helps allow the physiology to occur. So depolarization. Hopefully that rings some bells back from AP1, talking about neurons and skeletal muscle cells. Depolarization is when you go from the negative resting point to a less negative point, potentially a positive point. Now, depolarization here is rhythmic and spontaneous. Spontaneous as in the heart can do it on its own. It doesn't need anything else telling it to beat. Your heart is autorhythmic. Sure, the mind and your brain and everything else will get in there and help control it, but the heart by itself can still beat on its own. So that's why about 1% of your cardiac muscle cells have what's called automaticity. This automaticity is basically they self-regulate. They cause themselves to beat. They cause their own contraction. So that's why I put down self-excitable. Because they don't need anything else to tell them to beat, to tell them to contract or depolarize. So therefore, these cells, the ones that are self-excitable, can depolarize on their own, telling the cells around them, which are contractile, hey, it's time to contract. So these autorhythmic cells, automaticity, they happen on their own. Now, yes, they can be influenced by your nervous system, influenced by hormones, influenced by many different factors, but they can function on their own without anything else being necessary. And it's those gap junctions. Those are going to ensure that the heart contracts as a unit. Because you can't say, oh, let's do the left atrium contract and now the right ventricle contract. No, you've got to do it in series. Atria contract, ventricles contract. Atrias contract, ventricles contract. You have to make sure the blood is moving forward, not backwards. But what's interesting is, in comparison to the skeletal muscle depolarization, or even the neurons and their depolarization, the refractory period is much longer in cardiac muscles. Have down here 250 milliseconds. Now sure, that can vary a little bit from person to person, but it's pretty close to that. Go back to skeletal muscle, that was about a 4 millisecond range, 3 to 4 milliseconds. So cardiac muscle cells have a much longer contraction phase, much longer absolute refractory phase. So when this depolarization is occurring, we're still using the voltage-gated sodium channels. Now, hopefully remember what that means. Those voltage-gated sodium channels only open once a certain threshold is met, and then sodium floods through them. If you need a refresher, jump back to AP1, look at some of the nervous physiology or skeletal muscle physiology. The difference here, though, with this cardiac muscle compared to what we've already dealt with, is we're looking at a negative 90 millivolt resting state. The skeletal muscle, the neurons, were around a negative 70 millivolt resting state. So we've kind of changed where the resting state is. What we're looking at here is this depolarization is going to go down the T-tubules. The T-tubules cause the sarcoplasmic reticulum to release calcium. Same thing that happens before. So that is the same. T-tubules, sarcoplasmic reticulum, calcium being flooded in, that's the same process we talked about back in the skeletal muscle. But here's a new part. Slow calcium channels. The cardiac muscle cells have a second set of slow calcium channels to open. This is also why there's a longer absolute refractory phase. Because these slow calcium channels, they're not going to simply just dump all the calcium in. It takes a little more time. And as that keeps building, the contraction keeps occurring. Now, this goes through and creates what's called a plateau. Because as this calcium keeps releasing and releasing, you've depolarized, and you're kind of waiting in this plateau till you're repolarized. So this increased calcium amounts, this increased contractile phase, allows for this plateau to occur. So it's not the simple up and down we're used to. It's a little bit different. Now, this calcium influx, so we've already talked about calcium coming in, slow calcium channels. It's allowing this influx to occur, but eventually it helps out with this whole burst of calcium comes out in there. So you have this initial channels open up, these slow channels opening up. The calcium builds, and eventually it builds up to a maximal amount. And that calcium still is binding to troponin. So remember, troponin was part of the whole complex. Troponin, tropomyosin, actin. So it's still binding to troponin. And that troponin then will still go through a conformational change here. Now, the duration of the action potential, AP is action potential. That is much longer. As mentioned, that plateau phase. So it's really going to drag out more. Because as the heart's contracting, it has to have time to build up to enough force. Because when your heart is contracting, you've got to push that blood out into the bloodstream, into your vessels, which already have blood. So you have to be able to generate enough force to get the blood to go out to where there's already pressure. But then we do have repolarization. Now, repolarization can occur by the inactivation of the calcium channels. So therefore, no more calcium is flowing out. And as this occurs, the voltage-gated potassium channels are opening. This is going to allow the membrane potential to go back towards the negative state. So we still have the same basic idea of depolarization, repolarization, voltage-gated sodium channels, then voltage-gated potassium channels. But we've kind of changed at the middle. We've added this increase in calcium flow, we added this plateau phase. So it's the start and finish is kind of the same, but there's a difference in the middle, making it a longer process overall. So here's what it looks like. Now remember, if we were thinking about this back with skeletal muscles, with the neurons, this would have been, all right, fine. Start here at -70, go up, depolarize, and come back down, and we're done. But because we're now in cardiac muscle, we're starting down here at the -90 millivolts. We're still depolarizing based on the voltage-regulated sodium channels. But once we hit the peak, we're not simply just dropping back off. Instead now, what's happening is you have this entire plateau phase. Because the calcium channels are still open, calcium is slowly moving in. So you're still having contraction occurring. And that ends up with this very large absolute refractory period. We still have the small relative, but the absolute refractory period is what really has increased because of the plateau phase. Once you get through the plateau phase, now it's time to repolarize. That means the calcium channels are closing and voltage-regulated potassium channels are opening. And it heads back down just like normal, heading back towards the resting state. But because of that actual plateau phase in the middle, we've now pushed up into roughly the 250, 260 millisecond range. So yes, this starts at -90 millivolts, a difference, but then still depolarizes the same way. Plateau phase is different, but then still repolarizes the same way with voltage-regulated potassium channels. So a little bit of differences here, but yet some similarities. So taking a look side-by-side comparison, you'll notice skeletal muscle. Going from here, jumping up there, starting down at -85 in this case. It really depends on where that skeletal muscle is measured there. A lot of times we talk about -70. Then it's jumping up to +30. Cardiac muscle, -90 to +30. You then have a very short absolute and relative refractory. It goes up, depolarizes, down, repolarizes. But in the cardiac, we still depolarize the same way, then plateau, that's the different part, and then repolarize the same way. So it's still the same voltage-regulated channels. It's just adding that plateau phase in to make a longer contraction phase in the cardiac muscle compared to the skeletal muscle. So that's a little bit about how the actual cardiac muscle is built as far as the histology, and then how that relates a little bit to start with into a little bit of the physiology of the actual contraction itself.